Hey everyone, today we're going to be going over DNA structure. So that's what parts make up DNA, what is DNA, why do we care? So first off, here are the things that the state expects you to know and the things that you'll be tested over for your EOC. Last unit or last bit of information is we went over the cell cycle. Today we're going to be talking specifically about DNA replication. So this is your Learning Check 4B. I can identify the components of DNA and explain their significance. So, some of the things we already know about DNA. We already know all living things have it. We know it stands for deoxyribonucleic acid. We know from our biomolecules unit that it falls under nucleic acids, and therefore its monomer is a nucleotide. We also know from that unit that it contains carbon, hydrogen, oxygen, nitrogen, and phosphorus. We know that in eukaryotic cells, the DNA is in the nucleus, and in prokaryotic cells, it's free-floating all willy-nilly in the cytoplasm. We also know that the DNA contains the directions or the blueprints for how to make proteins. So think of this as like a cookbook. A cookbook doesn't just have how to make a vanilla cake. It has how to make a vanilla cake, how to make pies, how to make bread pudding, how to make ice cream, and then that's just the dessert section. Then it has how to make breakfast, how to make lunch, how to make dinner, assuming it's a huge cookbook, right? Our DNA is the exact same thing. It's got how to make all of the things inside of our body, not just one thing. It's got to know how to make all of the proteins and tissues for your heart, but also all the proteins and tissues for your brain. Also, in the last bit of notes that we went over that DNA is copied during S phase, which is an interphase of the cell cycle. So what is the structure of DNA? What does it look like? The shape of DNA is a double helix or a twisted ladder, kind of like this. If you were to cross your fingers, that's what DNA looks like. If we were to untwist that ladder, this is what it would look like straight. So we've got these these purple parts here, which are like the bits that we would hold on to if we were climbing up a ladder. And then we've got steps. Notice how the steps are half one color, half a different color. That's going to be important for later. So DNA molecules are shaped like a twisted ladder. We call this shape a double helix. Remember, words that are in bold, you're filling into your notes. The side rails of the ladder, so where we would put our hands on if we were climbing up or down a ladder, are known as the backbone, and those are made up of sugar phosphates. Remember, sugars are carbohydrates, so that's carbon, hydrogen, and oxygen. That's the CHO. And phosphate, that's that P at the end of CHOMP. We're only missing the N now. The steps of the ladder are formed by two nitrogenous bases, so that's where our N comes in, our nitrogen. So one here and one here. Notice it's two of them coming together. So half of the ladder is like this half with the side railing and the half step, and then this other side with this side railing and the half step. Together, these molecules form the side railings and the steps. They are DNA, and remember that the monomer of DNA is a nucleotide. So, just going back real quick over nucleotides, recall that they are made up of a phosphate group right here, a sugar right here, and a nitrogenous base. We could say a base, a nitrogenous base, a nitrogen base. All three of those things mean the same thing. There are, however, 4 types of nitrogen bases. They are adenine, thymine, cytosine, and guanine. Now, nucleotides, remember back here, they pair up, right? So we have to have 2 nucleotides to make a full step. This is that nitrogen base. That's this part right here. That's the half of the step. We have to have another half match up with this. The way that we do that is through the base pairing rule. Nucleotides always and forever pair up the same way. Adenine pairs with thymine, and cytosine pairs with guanine. One easy way to remember this is apples in the tree, car in the garage, or ATCG. Those are really the only 2. So how do we keep these pieces of the ladder together? Well, here is our sides right here, these blue parts where we would hold on to the ladder. and here are our steps that are connected. The sugar phosphate backbone, so here's the phosphate, here's the sugar, the sides of the ladder, they're connected by covalent bonds. Do we need to know what covalent bonds are right now? No, we just need to know that these are connected by covalent bonds from here to here. The nitrogenous bases right here were originally thought to be connected by weak hydrogen bonds. However, new research, like within the past couple of years, has suggested that it's not hydrogen bases, it's more of a polar and nonpolar attraction, just like the phospholipid heads and tails, how they push and pull against water. Same type of idea that keeps them together. However, for your EOC, you will need to know that these are hydrogen bonds. They're not, we've discovered that they're not, but for all intents and purposes of this test, you need to know that hydrogen bonds go in between the nitrogenous bases and covalent bonds connect the phosphates to the bottoms of the sugar. Now, the really cool thing about this is the order of these nitrogen bases determines what proteins are made and therefore determines your genetic traits. So let's say, for example, these were the genes that you needed for your eye color. So maybe TGAC is the order, the DNA sequence for brown colored eyes. And then maybe TCTC is the order for nitrogenous bases to make green colored eyes. So depending on the order of the nucleotides influences our genes and how they are expressed. And that's it for DNA structure. The next lecture will go over DNA replication.